All right, so welcome everyone. I'm Nicolas, I'm a senior application scientist at BDB Informatic, former Flojo. Here's my email, nicolas.love@bd.com, so don't hesitate to reach out to me if you have any question. Okay. So we're gonna talk about Autospill today mostly, but before, and composition in general. But before we jump, I want to make sure we're all in the same term of the wording. So we're gonna go through glossary. Then I'm gonna present you traditional composition, the composition you probably know already. And I'm gonna show you talk about autospill, the new way to do a composition. So let's talk about fluorescence first. What's fluorescence? It's a rapid absorption of energy and the emission of that energy after produced light. And we use a fluorescence dye to mark our antigen or antibody, and we're gonna be able to target a specific markers on our cells to detect those markers. And with that detection, we will be able to estimate the abundance of that marker as well. So each dye have an excitation. Here, for example, for PSI 5.5, it's around 560, a yellow-green laser, and you have an emission spectral. Okay. And here's the maximum is around 675. And we use a primary detector to detect this dye into the instrument. Okay. So the primary detector usually have a bandpass filter. So it's around 670 here to 690. So probably 570 plus or minus 20. A bandpass filter, and that's where we're gonna the photon that have energy between that line to that line will be catch by that detector, and it produce a voltage, and we will see after that voltage transform in electrical pulse, and you will see that on your instrument, a dot in your instrument. But as you see, the emission is not straight. You have other detector, and those called a secondary detector, and they will catch part of the photon also emitted by that dye. And that signal detected by the secondary detector is called spillover. So when we create a spillover matrix, okay, it's basically removing the spillover of that dye to every secondary detector. And traditionally, spillover matrix show a percentage of relative to the primary detector. So we have 100%, of course, in the primary detector, and we hopefully get a lower percentage on the other detector. So a lot of word about unmixing. So unmixing, basically, it's a specific, a composition is a specific type of unmixing. Okay. So when a sample is a full complement of dye, they are carefully stained. The light detected by each detector is a combination of emission from all the dye present of the object being interrogated. And mixing is a process of estimating the abundance of each dye and for each cell. The estimate should add to the signal observed for each detector. So unmixing is the estimate of the abundance for each dye for each event. So what's composition? Okay, composition is one particle on a mixing algorithm, as I mentioned earlier, and so invert the spillover matrix and multiply it by the data. Sorry. So composition word come at the beginning from the idea that you're fixing the spillover, and if the spillover is something wrong, but the spillover is just what it is, and we just try to deal with it. So there's only algorithm for mixing when you have one dye per detector, and it's called the traditional cytometric case. So how you estimate the spillover? You're gonna run a single color control. So you're gonna run FITI only, P only, or whatever, And you're gonna tell the machine I'm running VT only. So here's the signal that you should see on my VT. If you see any signal on any other detector, that's coming from my spillover, and just remove that signal. That's the percentage of spillover that I'm getting. There's 4 rules of to have a good compensation. The first rule is to make sure that your compensation control fluorochrome match exactly the same as the one from your experiment. So if you're running a FITC, you should have a FITC compensation control, and you cannot use a GFP as on your cells and FITC on your compensation control. Those two dye are totally different on emission, so your compensation won't be accurate if you do so. It's also true if you use tandem dye, for example, so PSI7, APCH7, or things like that. Those dye are lot to lot dependent. The fluorescence change a lot from lot to lot. So you need to make sure that you use the exact same lot number for your composition than for your sample. The second rule is to make sure that the background fluorescence should be the same for the positive and negative control. That means if you use cells for your composition as a positive, you need to use cells for negative, and vice versa. If you use beads for positive control, you need to make sure that you have beads for negative control. Okay, doesn't mean you cannot mix match in different tubes. So you can have tubes of cells and tube of beads, and for your composition, and I'll show you that into the demo. The third rule is to make sure that your control is at least as bright as your sample. Okay. So the MFI, mean fluorescence intensity, of your composition control should be at the same MFI at least. That's your sample. And the fourth rule is to make sure that if you do any treatment after staining on your cells, your composition need to follow the same treatment. So the most common treatment used is fix and perm. So if you do that on your cells, you should do as well on your composition control. If you just do fixation, same thing. All right, so just traditional and there's a QA. Do I need FMO control for composition, or is single state control sufficient? So FMO of fluorescence -1 control are not for composition. FMO are gating control, is to make sure that you put your gate at the right place. So just for composition purpose, it's only a single stain control that you need. All right, so traditional composition, so you have detector and you're gonna remove the percentage. For spectral mixing, same thing, you have just get the full spectral, but the same idea. You're gonna get a spectral matrix instead of a spillover matrix, but at the end, you still want to know the spectral and mixing matrix compared to the composition matrix. But in FlowDraw, you won't have any difference if you use traditional composition versus a mixing composition. All right, so let's jump into traditional composition. Sorry, we have a question from team. Should I perm and fix my composition beads if I've perm and fix my cells during the staining? Yes, you need to do the same treatment because your dye might be affected by the fix and perm, so your emission may change. So you have to do the same treatment on your beads than on your cells. So another question for Adil: for each sample, when you see FITC, PE, and it gives the percentage of each channel is splitting into the other. Yeah, so composition is exactly that. For here, you have 12%. If on FL1 is your FITC channel, FITC is 100% detected. We consider it's 100% detected in detector 1, and it will be 12% in detector 2 for the FITCP channel, for example. and FITC will be FL2 will be 100% here for the P detector and 15% into the FITC detector. Correct. All right, so traditional composition, the one everybody use or used to use at least, that you find on any acquisition instrument for sure, and in most analysis software. So the first thing you need to do when you do a traditional composition is create a cleanup gate to set up your autofluorescence. So here's my cells right there. There were PBMCs. I select lymphocyte. And you need to make sure that this gate for the cleanup gate focus on the population which express the antigen targeted by the probe. Okay, so if I'm looking at CD4, CD8, or CD3, I need to select my lymphocyte. If I look at my CD14 monocyte, I need to select only my monocyte. I will not do a huge gate to select everything. Not my lymphocyte, monocyte, granulocyte. I won't do a big gate because your autofluorescence will be different, so you will affect your composition. Okay. All right, so then you're gonna, after the cleanup gate, you're gonna have to do a negative gate and a positive gate. Okay. And the software will calculate the mean fluorescence intensity for your negative population and your positive population. Then it will calculate the spillover between this population. Okay. So the mathematical calculation is mean fluorescence intensity for a P and minus the negative and divided by the 780 here negative. I don't have the slide showing that. I have it somewhere in another presentation. But here, it's how you calculate the spillover. And it's just calculating here how high is and how much percentage I need to remove to bring it back to the same level as the negative. All right, so if you calculate the spillover properly, at the end, your mean fluorescence intensity for the negative cells and the positive cells are equal for the secondary detector. All right, so just get positive and negative event on each single cells control. It's easy, right? And that's what we could think, and that's maybe why you like, okay, composition, no problem. But it may be also difficult to achieve, as you may know. That's probably why we have 100% following that seminar today. So why we have the issue with composition? Can be because we have a low frequency expression. Okay. We can have also low antigen density expression, and we have also autofluorescence. Three things that make it difficult. All right. So if we have a low frequency expression, I've for here an example only 0.72%. So really low number, not enough cells to calculate my composition. The low antigen density, that means my MFI is too low and really have really few cells, and autofluorescence, my negative, just a shift, and I cannot really see my cells, or too bright to see my cells. So if I don't have enough frequency or low antigen density, I kind of, the idea of a lot of people is just to move my gate to get more cells and to get the 2% minimum that is required normally to calculate the composition. But moving this gate doesn't make it better. You see the other way, you make it worse because your MFI shift. See the plus sign here shifting, and it's getting really close now to your negative. That means this MFI, that's going to be the MFI for the calculation of your compensation. That means your positive sample should not be brighter than where this plus sign is. And of course, it won't be the case because you have your some of your positive cells right there. Okay, so that means your compensation won't be calculated properly. So we're going to talk about Autospill now. Autospill is a new way to calculate compensation. It has been published in Nature, so you can look at this paper. So Autospill, a method for calculating spillover coefficient in high parameter flow cytometry. And the idea of Autospill, instead of calculating just the difference, forward, instead of calculating the difference here, the spillover, is going to take in principle that the percentage doesn't matter. Your brightness of your signal is always the same percentage. So it's always 15% of FITC bleeding into P, for example, or it's always, so it doesn't matter if your signal is at 10 to the third or 10 to the fourth, it's still the same percentage. So we will be able to find the slope, okay? And this slope is the compensation slope, and we can calculate or remove that slope and bring it back to a 0 degree slope. And to do so, we're going to create a linear fit, okay? And it's a robust linear regression. And we won't have to do a positive gate or negative gate. This algorithm, Autospill, will automatically create that gate to fit our sample. And when you find that gate, as I say, it will bring it back to 0. And as I mentioned a lot, it was a robust linear regression. And the idea of this robust linear regression is because it's going to find our cells and everything that's kind of outliners that when you do traditional compensation data included here for Autospill, they won't take in consideration, or at least they will weight less into consideration to draw our slope for the compensation. All right, so when it did find the slope, it's going to correct it, it's going to compensate it, it's going to refit the data to estimate the error, and it's going to, if find any error, it's going to calculate a new fit, so a new slope again, and it's going to repeat the correction over and over until it doesn't find any compensation to correct. And also the beauty of Autospill is you can remove autofluorescence. Okay. So autofluorescence is just a signal, a fluorescent signal, like any fluorescence marker, but it's just coming directly from the cells. So it's gonna give you a signal in every detector. And so this signal is gonna create a slope exactly like a fluorescence detector, and Autospill is gonna be able to find that slope and correct that autofluorescence from coming from cells. To correct that slope, you will need to open a new detector. So you need to have an empty detector that you're not using for your experiment. So if you don't have, if you don't have FITC or if you don't have V450, for example, you just still open that channel into your instrument, you leave it empty, you don't have any you don't have any fluorescence coming from your cells or from your staining on this detector, but we will use that detector as an autofluorescence detector to detect the signal coming from the autofluorescence. And by using that detector after in Autospill, we will be able to remove the autofluorescence. And that's where the cleanup gate play a big role in Autospill, because you're gonna be able to create a multiple cleanup gate based on your cells, and you will be able to create multiple matrix based on your cells. So of course, your compensation of fluorescence composition won't change, but the fluorescence composition may change between the cells or will change between the cells, and you will be able to correct your autofluorescence based on your cell population. All right, so what about spreading? So usually the traditional way to calculate SSM or spreading matrix algorithm depend on a well-defined positive and negative population. But we lose that by that's one of the benefits of Autospill, and we're losing that by using Autospill. So we had to find another way to calculate the spread. So the solution, we partition the data into, yes, Javier, you raise your hand. Go ahead. I don't know if I have to. Can I ask question? Where are you? No, I don't see you and raise anymore. I have a question in the chat. Do you need to make sure the channel that you choose for autofluorescence does not contain spillover from other dye used in the experiment? Not at all. You can open any other channel, doesn't matter. Usually you try to find a channel where you have a lot of autofluorescence. So something that is excited by the usually the blue laser, the violet laser, or the UV laser. The most preferred detector. And usually, so that's the excitation and emission, I will say below 550. Let's maybe a little bit, we push a little bit to below 600 in the emission. You will detect the best autofluorescence. All right. So the solution for autospread, to calculate the spread with the autospill algorithm, is we're gonna partition the data into quantile. We're gonna estimate the fluorescence in the primary detector. Then we're gonna estimate the standard deviation in each secondary detector. And then we're gonna calculate a linear fit to determine the standard deviation at 0. Okay. So we're gonna shift that, bring it back to the 0. There you go. Then we're gonna adjust the data. We're gonna calculate the linear fit to determine the spillover spreading coefficient. And so for the with that, we will be able to calculate the SSM. So okay, and we're gonna call that the auto spread producer, a spillover spreading matrix that barely deviate from the traditional algorithm. So here's this SSM produced by Autospread. All right, so what's the advantage of Autospill and Autospread compared to traditional composition? Well, first, it's less demanding because you don't need to do any gating. Okay, the only gating you need to do is your cleanup gate. You don't have to define your positive and negative population. So you don't need to manual tune your spillover matrix because the algorithm is gonna automatically recalculate the compensation, make sure that the compensation is as good as possible. If you find any issue, it's gonna automatically adjust it. And because it remove the outliners, or at least weighted them less, the fit of your composition should be way better than with a traditional composition where any outliner we have the same weight as a regular sample. And also the advantage is, of course, you can remove the autofluorescence if you have an open channel that you haven't used. And as I mentioned, works-based when the channel has a high level of signal for instant control. All right, so of course, you're gonna have some limitation also using Autospill. So you need is perform a linear regression, and usually to perform linear regression, so to find that slope, it's better to use cells than beads. Doesn't mean beads not gonna work. It just beads gonna probably give you a really similar data than traditional composition. So not a big advantage in let's say except speed, and because it's way faster to run Autospill than traditional. But except speed, the value of your composition will be the same, and the goal of Autospill is to get a better composition. So some limitation there. So Jana asks, can you tell us the best channel for autofluorescence for lymphocytes? I will say anything in the UV or violet that you have a channel open. So try V450 will be or Pacific blue, depending on how you call that detector, but violet excitation 450 emission, or even a violet excitation of 510, 530 emission should be pretty good. Same thing on the UV excitation around 450 should be great. I will say you can go in the blue laser. Blue laser you will fall into the VC detector maybe, because after that below 600 in the blue laser, depending if you have a yellow-green laser, but you may have the PE detector there as well in the 580 something that you can choose. But my first choice will be violet or UV laser and around 450, 530 detector. So Daniela asked a question. So basically, we need to leave a dump channel in order to perform this autospill in all staining. Are you going to show a live example on how to detect it? Yes. Yes, I will show a live sample. And it's just not, it's not a dump channel. It's just an open channel that you don't use. Yes. Couple question. I can autospill be done in Diva to see what's the composite data to acquire and make sure it looks good. No, autospill can not be done in any acquisition software, so it's only on FlowJo so far. This cannot be applied to previously acquired data if we did not have an open detector. Yes, if you didn't have an open detector, you cannot remove autofluorescence on previous data. If you have cells and beads, what do you recommend? So for autofluorescence, it will be cells. You need to have an unstained cells sample also. Okay, so you can have your composition on beads, full beads, but you need to have an open channel and an unstained sample that you will run to remove the autofluorescence. All right, so no more questions so far. Okay, so let's go back. So limitation of autospill. So of course, you don't need to fine tune, because normally with the fine tuning, you should not have to do it anymore. And as I mentioned, autofluorescence, for one of the questions, autofluorescence should not be estimated using beads control. So you need to have unstained cells to be able to find the composition and remove the autofluorescence. Sorry, you should have an unstained cell sample to be able to remove autofluorescence and an empty channel. All right, so more questions popping up. For FMO, a sample can be used beads. No, for FMO, you need to use your sample. FMO stands for fluorescence -1. That's not a composition control, that's a gating control. So make sure you put your gate at the right place. So if you gate on CD4 on your FMO, you have all your fluorescence except for your CD4 marker. So you can put your CD4 gate to have a 0% cells into that gate. So when you go to your full stain sample, you make sure that everything that's shifting and coming to that gate is really CD4 positive. All right, so let's see an example. One more question for autofluorescence. Do you need the same number of cells as for your specific stain tube? No, you don't need the same number of cells, just a regular number of cells. So that's no problem. And Karina, you ask, is it possible to speak? Yes, normally if you raise your hand. Oh, there you go. Sorry, I didn't see you raise your hand. There you go. You're permitted to talk. Go ahead, ask your question.
Okay, thank you. Now, my question regarding bits and cells, it was, I wanted to know what do you recommend if we do the automatic spill, auto spill? It's because quite often, well, I would like to use cells and bits sometimes. Because sometimes I have recombinant antibodies. I cannot use bits, at least the Becton Dickinson. So I have both, but then if you suggest that the autospill works better with cells, but then I have bits at the same time.
That's fine. So you can have a few sample with beads of a Fleur marker with beads, but most of the marker with cells. So for the one that need to be used on beads, you just keep the beads and you do cells on every others.
Yeah, but then when you do the compensation, when you use the auto spill or the auto.
I can show you that on the demo.
Okay.
In my demo, I have cells and beads, and I can show you that. You just define.
Okay.
Okay, so I'm just gonna read. So answer that question. The number of cells. We answer that question also. When working with complex tissue made of different cell type, do you make different composition matrix for different cell type? That's from George. Yeah, so you will make a different cleanup gate, and it will give you a different composition matrix based on you want. If that will be the only way. One way that is not always feasible, because you may want to focus in your analysis in different cell type on the same tube. So one way to do it is to select the most autofluorescent cell type to remove. Okay. I see some hand raise. As cells, you can talk. No, not talking. Okay. Look, what else? Let's Paul. Paul, you wanted to talk. I just gave you authorization.
Yeah, I was just wondering, auto spill values are similarly to like the traditional compensation. Are there values that are too high that you would want to, work on your panel a bit more? Like, with traditional compensation, you try to keep everything under 100 or something like that, some value.
Yeah, so the compensation value, the percentage.
Are there values that you're worried about with autospill?
Yeah, so even for traditional, so the percentage value is not telling you really if it's a good or bad panel. It's the SSM who's gonna tell you if it's a good or bad panel. But yes, Autospill is not gonna prevent you to do a good panel to get good compensation. It's just gonna try to optimize your compensation and not having to manually adjust your compensation. So the value, when you have 100% of compensation, depending of how many color and how many, what's your instrument capacity. But if your instrument capacity is a 15 color instrument and you use 15 colors, of course, you're gonna have huge compensation between some channels. If you design your panel properly, doesn't really matter that you have a huge percentage, and you don't have any choice because you don't have any open channel, but you make sure that you still can separate your positive and negative population. So Autospill won't, let's say, there's not a number neither for traditional Autospill saying if you have 100% or it's not a good panel. You just want to make sure that you're able to define all your population the way you're supposed to.
All right, thank you.
gonna lower hand because same, and I have another question here. So it is redundant, but we will acquire the data uncompensated, but also acquire a compensation control at the same time, and then do all the compensation calculation in FlowJo after. Yes, or you can just do the compensation on the instrument if you want to be able to kind of see the data when you're running on the instrument. It will be just traditional compensation on it. And when you go in FlowJo, if your compensation is not the best, you can just run Autospill on it for sure. All right, so here's an example. So that's the example from the paper they published. So it was a traditional composition. They were losing their Treg population, the CD420, CD4, CD25 double positive population. And when we look into the data, the reason is there was a composition issue, the CD25, CD19, and so they were losing that population with the gate CD19 TCR beta, and the population was out of the gate. Okay. So of course we could have tweaked the manual the traditional composition manually to try to compensate that or make sure that it was fitting, was at the right place. But instead we just ran Autospill and the same composition control. We didn't change the composition control. We didn't do anything manually. So we just ran the algorithm, and the algorithm was able to recover that population Treg, if it was at 935% here. And you see that the composition for CD19, CD25 was done properly, and so our population was back to the where it's supposed to be in the CD19 TCR beta. Okay, another question pop up. How are you able to do traditional composition at the machine with an open channel with no signal? You can just tell the machine to ignore that signal in the composition. In Diva, you can open all the channel will be default open, but you can select one and say remove that channel in Diva. If you're using beads, will the Autospill work better if the doublets are included? No, William. If you're using beads, basically the Autospill is gonna give you the same number as a traditional, or really similar. And we'll see in the training, because I have mostly beads that are for composition, so we'll see that the value are really similar. But it's you just funny at the field evolve. You know, we were doing composition with cells like 15 years ago, and we all switched to beads because we had better composition at the time. And now with Autospill, we kind of go back to cells to have a to solve all the issue we have, like the one here on top, where sometimes beads because they're not perfect. And they are different than the cells. We don't get the right composition. All right, so how to do Autospill in FlowJo? So you just do the same thing. So FlowJo Autospill is available after FlowJo 10.7, so 10.7 and after. We are currently in version 10.8.1. So if you haven't upgraded yet, I highly recommend to upgrade to the newest version, so 10.8.1. It's free of charge, so you just can go on the website, download, and the new version on your computer. So when you open FlowJo, you select your composition group, and you click on your composition, and in version 10.7 or after, now you have the option of selecting either a traditional or Autospill Autospread. Okay, so you just select Autospill Autospread, and you click on next, and you will run the composition. Okay, that's just these two options between traditional and Autospill. All right, so spectral. You can also perform a spectral composition to to do compensation. Okay, so if you have a spectral instrument, you just select spectral, and if you select spectral, you can do both autospill or traditional. Doesn't matter. It's really up to you on basically on what type of instrument you have. So yeah, as I just so your question. Yes, you can select spectral with autospill. There's no problem. But if you don't have a spectral instrument, don't select spectral. Only select spectral here if you have a spectral instrument. You can also do spectral if you want, but it's not full spectral. But you can do spectral on opening every channel of your instrument. Even the one that you don't use, you open your, I don't know, 15 parameter instrument, you use the 15, even if you're running only a 12 color experiment. And that's spectral. It's not full spectral, but it's spectral. And that by selecting, by opening all the channel, you will be able to collect more signal for the low expression target. So we'll be able also to maybe detect a dimmer population. And the last option that you have in the FlowJo is optimize weight. So running optimized weight function for a spectral composition will basically try to minimize the SSM, the spillover spread matrix. And it will weight the different detector. I will try to find the weight on each different detector to minimize the spread. Okay, so it will a big issue with a spectral instrument is your negative population gets spread a lot, quite a bit. And with optimized weight, we're gonna condense, or we will be able to condense your negative population. So your dim population will be more resolved than if you don't do that option. On a special note, when you optimize weight function, so don't do it every time, do it only for samples that require it, because that require a lot of computing power. So when you click on that button, you may have to wait a few minutes, maybe even more, to the algorithm to calculate the weight for each channel and minimize the spillover spread matrix. All right, so let's jump into the demo now. Okay, so question, have there been comparison done between conventional compensation versus semi-full spectral, just using old channel or the conventional? Okay. Yes, there's comparison done, and you have a better detection. That's that's one of the big advantage, I would say, of spectral is you get better detection for a dim population. But as I say, usually there's a trade-off, because even if you get better signal, you have also more spread, so it kind of cancel each other. So, but yeah, you get more signal on the dim population with spectral. Okay, let me share my screen. That. All right. Let's open FlowJo 10.8.1. So in the newest version of FlowJo, I don't know if you're aware of, but you have a button, remember me, so you don't have to type your password every time if you check that box. And if you don't, to get that option, you just go in the preferences, license, and in the license, it says show the button, remember me. Okay, by clicking that, it will, when you pop your portal login information pop up, you have to remember me, you just have to check. Another function that I like to mention but it's not the topic of today's talk, is the enable autosave. So select that and then put a time. So that means every 5-10 minutes, whatever time you put here, your workspace will be saved. So if something happened to your computer, if it crashed or if you restart because IT push an update or upgrade on the computer, you will lose only the 5 or 10 minute interval max. All right, so let's take some data. As I say, I'm sorry, I don't have much data available to show. So here's an experiment. Guys, if I remember correctly, is a so it's an 11 color experiment. Okay, I have FMO, I have my sample. I'm gonna create different groups, so all my composition are already there. We create just a group FMO quickly. Base one. Create my group samples. Are you? Yes, they are PBMC currently. They are human PBMC, exactly. This was a plan to add a what do you mean by OLS? When you variant of unmixing, we already have unmixing into FlowJo. I don't know what you mean by OLS. So when you do select spectral, you're unmixing. That's ordinary least square. Don't know what it is, and so I cannot answer that question. Sorry. All right, so let's do composition. Select my composition tube as a group. Click on composition. It will create my cleanup gate. Okay. So we will do traditional first. I'll show you. Yeah, let's do traditional. Traditional first. Okay. All right, so it did a cleanup gate. As you see, it's mostly beads. I have only a few cell sample, 1 CD3 cells, unstained, and my live dead marker on cells. There's another question. When you put a single stand control with cells instead of beads, how do you process with a rare marker? Example CD3 and CD4 is abundant enough for positive control, but how do you make sure you have big enough for positive control to rare event like CD25 or CD19? Just record as much cells as possible. That's, I would say that if you can already purify your cells with some kit to purify your population, that's another way to do it, to already kind of select your CD19 cells with beads or thing like that. But you should have with those markers CD19 CD25 as you mentioned, you should be able to have enough cells, positive cells, to just do it directly from PBMCs. All right, so let's first things, traditional. I can choose the color. I'm gonna leave it on red. Okay, so it's not spectral data, so I'm gonna not check that. Select traditional. It's good there. All right, so make sure that the parameter of my machine is the right tube associated to that. So if it's green, that means we find the name of your tube in it. So here, RD, define it, the PCH7 is fine. Alexa 488, it's labeled A48, so it's fine. My Alexa Fluor 647, that's my blank. That's the one I'm gonna use to remove autofluorescence. So I know you heard me talking about using violet laser, but this one, that was the only channel available, so it was run on the red laser. But for traditional, I cannot remove autofluorescence, so I'm just gonna click on that and remove this parameter. Okay, so my 647 disappear. Then Alexa Fluor 700 for CD3, PE for perforin, PSI 5, PSI 7, everything else is correct. Okay. Now I need to set my universal negative. Here's on cells, and that's not correct. I need to put on beads everywhere. Most of my samples are on beads, so I'm just gonna put beads everywhere at the beginning, just as a start. All right, so now I did select my negative, and I'm gonna go to my compensation. I'm gonna start by the bottom, and you see that's my universal negative for beads. That's my positive here for Pacific blue, and that's my cleanup gate. Let's select the bead, so I'm selecting the same thing. That's fine. Okay. If I was not, I can always select size for the beads and put it in all the compensation if I want, but that works here. All right, you select my positive for Pacific blue. I'm at the right place. The blue is my negative for Pacific blue. All good there, nothing to change. The Texas red, same thing, negative, positive, all good. PSI 7, negative, positive, select the positive, all good on beads. PSI 5, same, that's correct. PE, same thing, correct. And now we arrive to my Alexa Fluor 700. And you see my Alexa Fluor 700 was done on cells here, okay? And my controller is on beads, so that's not correct. That don't follow one of my composition rules, where my negative and positive should be on the same particle, which is here my cells. So I'm gonna remove my universal negative on beads, and I'm gonna put my universal negative on cells. Here's my universal negative on cells. Okay. Can make sure. So here, see that also my gate for the positive is not correct. It's selecting everything. It's selecting also my blue peak, which is my negative one. So I'm just gonna double click and select only the positive cells. All right, so now I'm good for Alexa Fluor 700. I have my negative, positive peak. Let's go up. Alexa Fluor 488, good as well. APCH7 is fine. And we get to my RRD, which is my live dead marker. Same thing is on cells. Here, my universal negative is on beads. I could put my universal negative on cells if you want. Let's do that just for now. And as you see, the positive peak, I don't see any positive peak here. And we know that our dead cells usually have a low forward scalar and a high side scalar, so they're probably here on top. So I'm gonna change my gate here. Okay. I'm gonna include a little bit more dead cells, and you see here if I select here on top, I'm getting some dead cells signal. Okay. I want to select a little bit more again. All right, so now I can adjust my yeah, there's a question. Do you have a recording of this webinar? Yes, this webinar will be recording and will be put on the website. So now I need to select my positive population. Here's my positive population. Okay, 12% of dead cells. Okay, good. All right, but now if you look at my negative, you see the blue line is my universal negative. my green line is my negative for my sample, and here's my positive for my sample. And you see that my negative are shifted. Okay, that means the autofluorescence between my sample and the universal negative is not the same. So that means the autofluorescence between my positive and the negative I'm taking will not be the same. So if I run my composition like that, I will not have a good composition. So to have a good composition, I need to define my negative. And the way I'm going to define my negative, I'm just going to define it by using my ARD sample, but by selecting the negative cells. And I'm going to call that the negative, create a negative gate here. Okay. And now instead of using my universal negative cells, I'm just going to select my negative cells from my sample. Okay. So I have my negative and positive cells. And that you can do for every sample, not only for live cells. So if you have a mix between beads and cells, and your universal negative is only on beads, and you don't have a universal negative on cells, or vice versa, you can select your peak on the specific on the same tube. Okay. Usually that's why composition beads usually have a negative peak and a positive peak. So you can select that to flowchart. All right, so when everything is defined properly, click on View Matrix. Are my beads BD? To be honest, I have no idea, Carolina. It's not my data, so I don't know what beads they use. All right, so click on View Matrix. I have my composition matrix here. Okay, and now you have we have the percentage. Okay, I can see my matrix on a specific sample. So I'm gonna take, for example, this sample, which is double stain, double stimulated sample. So I should have signal on everything, and I can select a specific population if I want. Here I didn't start getting my population, so I don't have any population. But if I did start getting, I could see my forward scatter, side scatter gate, my doublet discrimination gate, and select only for the cell population I want to see. The blue, the light blue, it's my uncompensated data. So the same sample, but uncompensated. My black dots are my compensated data. If I don't want to see my uncompensated, I can just uncheck that, and I see only the compensated data. All right, so my composition matrix is done. So now I'm gonna check to see if it looks good. So it's only by eye, so it's not really accurate. But what I want to, what I don't want to see, I don't want to see any banana shape or hockey stick shape form. So either that means my positive cells going either from going in the inside or going to the outsides. If something looks quite square, guessing it's pretty accurate. So square, square, everything seems to be square. So my composition looks pretty good there. Okay, so that's how you do traditional composition. Now I can apply my composition to my sample. To apply my composition to my sample, I click on the M here and I click and drag that. Okay, I can click and drag that to a specific sample if I want to change only the composition of that sample. If I want to change the composition of a full group, I can click and drag that to a full group. I can apply to all my sample if I want. Okay, so if we go on my samples here, you see that I have a matrix. So here the square, because you have a gradient inside, that means there's a matrix, a composition matrix that's already been applied. If I put my mouse on it, it says it's a acquisition defined. That means the one that's been calculated on the machine. Okay, so on I think it was run on the Fortesa, so on the Fortesa. So I can have it here as well. So that's a composition calculated on the Fortesa. If I want to modify that composition for that sample, I click and drag and put it on that sample. I want to click and drag that on all my sample, it will be applied to all my sample. And this grid become the same color as your composition. All right, so let's do Autospill now. So I'm gonna close that, go on the Autospill. Okay. Won't be able to start from scratch, but I'm gonna have to put it back to where we were. Okay, so we were like that. All right, so that's how we started ish. This one I'm gonna call Autospill composition. Okay, so same thing is not spectral, so I'm not gonna select that. I'm just gonna select the Autospill auto-spread. Okay, same thing, I'm gonna make sure all the detector have the right tube associated. So RD, H7, etc. Here I have my 647, which is gonna be my autofluorescence removal. And make sure that I have my unstained cells here. And everything else was correct. So as I mentioned in the slideshow, we don't need universal negative, or we don't need negative gate, neither positive gate. The algorithms automatically gonna define those populations. So I need to remove my negative, and we have a button here to remove all the negative. Okay, so we remove all the negative population. We don't care about the positive. The algorithm's not gonna take into consideration the positive gate right there. Okay. Okay. So my cleanup gate is done already. I don't have to adjust it. If, of course, you will have to adjust it if, for example, to make sure that you select the right population to be where the marker is expressed, because everything is already accurate here. I can just click on View Matrix. So you see way no gate adjustment, nothing, so way faster to run. Just have to make sure your cleanup gate is at the right place, select Autospill, and that's it. You remove your universal negative and run Autospill. Okay, so here's my Autospill matrix. So as you can see, pretty square as well. Very comparable to my traditional, which is kind of expected, as I said, because it's beads. So if you use beads, it will be really similar to Autospill will be really similar to traditional, because beads they are really well defined positive and negative. So the way the algorithm is going to calculate the slope, it's just going to kind of draw a line between the positive and the negative. Compare that with the cells, it will be easier to find the slope of the cells. So here's my matrix. Same thing, if I want to apply it, click and drag my hand to my sample. So I apply it on my FMO. So my FMO have Autospill, my sample as traditional, or I can apply that to myself. All right, so let's say you're trying Autospill for the first time and you want to compare your Autospill with your traditional to see if, what changed, what did better. Before we go there, before I jump to that, you can see that here on my Alexa Fluor 647, I have value of 39%, 30%, etc. Here 28, 25, 33, 34, and same thing on the other side. I have composition, and that's my autofluorescence here. This line, that's my autofluorescence removal that I didn't have in my traditional. Okay, so now let's compare my Autospill traditional. Of course, I won't be able to compare the autofluorescence because I don't have it in traditional. But if I select my two matrix here, I will see the two matrix overlay. Okay, and here the value that I'm seeing here is the difference between Autospill and traditional. Okay, so as you see, the value are pretty similar. I have only minimal variation between those two numbers. As I mentioned, it's because of beads. So if it was cells, you probably will have way more differences, but because it's bits, it's really, really similar. The big advantage that I have right on this sample to run Autospill compared to traditional is that he removed my autofluorescence. Okay. All right, so that's how I ran my composition with Autospill. Carolina, you can talk now.
Yes, thank you. Sorry, can you repeat something? Is when you have the cells, the unstained cells for the autofluorescence, you have chosen any a gate for that. For example, if you have the PMCs, if each T cells, monocytes, neutrophils will have different autofluorescence. So I could not understand exactly how you did it, because you take the whole sample, or you take just the gate that we are seeing in the AARD.
Sure. So to remove autofluorescence, here what I use, where's my unstained cells here? So here my unstained cells, I just select my lymphocyte right there.
That's the question, because then the thing is, what is going to happen with the other population? In fact, I am analyzing samples from patients where I'm analyzing the 18 parameters, having T cells, monocytes, and neutrophils. And in fact, I have to run a compensation for every group of samples. They are analyzing the same panel.
So as you see in that panel, the goal was really to look at the lymphocytes. That's why I selected the lymphocytes. So that was really the focus on that analysis. If you want to, if your goal is to analyze monocyte or granulocyte, you should put your gate on that cell population here.
No,
for
I will not
for the autofluorescence.
differentiate. That's the point.
So if you
In
want
fact,
to
the whole
select,
panel is the whole group of cells. I mean, 5 populations. So
yeah, so right now the only way will be to select the highest autofluorescence population. So
okay.
we'll probably to try to remove that autofluorescence on that cell population. It will not affect too much the other cells, because if they don't have autofluorescence, they will have almost no correction. So I will select the trouble
highest.
population. Yeah.
Okay, excellent. Thank you.
Trying to see if I have any data that I can show with autofluorescence. I don't think so that I have anything available. So Luran, you asked, does it mean that we have to set 4 compensation matrix if there's 4 population? No, you cannot set 4 compensation matrix, because your compensation will be applied to only your tube. So if you have 4 population in it, you cannot apply 4 different compensation matrix on the tube. So as I told Carolina, you will have to you will select the brightest, or the one that gives you the most autofluorescence. If you have spectral, that will be kind of the similar as well. When doing autospill, and since you don't have to fine tune the population, it's just better to expand your size gate to include all cells. No, ideal. No, you need to select only the cells that are positive for your marker. So the way Autospill will work to find your cell population, it will just take similar cells or similar cell size. So you need to make sure that your cleanup gate is tight around kind of the same type of population. So not recommended to not doing a cleanup gate. Autospill, when you choose to remove universal negative, you still have to manually mark the negative path for each control, isn't it? No, trying to, let's see if I try to understand. So when I run the compensation here, add here my universal negative for bits or whatever, because it created automatically. And the only thing doesn't matter. The only thing I had to do is select Autospill, remove the universal negative, that it will automatically delete on my universal negative there. Okay, I still need to have a cleanup gate, which is different than my universal negative. I still have a cleanup gate to say where are my cells here, which in this case were my bits. All right, so just a comment. Didn't know you can show difference between the two matrices recall. Thank you, CK. So yeah, you can, any matrix, you can select any compensation matrix and see the differences between the different compensation matrices that you've done. Okay, so for example, if I want to compare the traditional versus the one I ran on the machine, I can double select those and it will show me the differences. And in theory, you should have no difference between the two traditional, the one done on the instrument and done on a flow draw. The only difference may be where you put your gate, and you may have your MFI might be slightly different, which may give you slightly different compensation. That's why we see Alexa Flow 700 and RRD, which are the two cells base, because I adjusted the gate manually. That's probably why you see a difference there. Roma, so it's not good enough for a mixed cells pop to just draw a big region rather than choosing specific. Yes, if you want to remove autofluorescence, it's not, you cannot draw a big region. It's recommended to select a tight population, yes. So from George, sorry, I might have missed it. Are there recommended fluorescence wavelengths for autofluorescence? Autofluorescence, usually violet laser, maybe UV violet laser, blue, and with a maximum emission around 600 maximum. Any channel. So basically, usually for most of the cells, the lowest excitation as possible in wavelength. So 355, 375, which are both UV, 405 for violet, 488 for blue, and the emission of the light emission between whatever your laser is giving you, so the wavelength of your laser all the way to 600. But as you see, I did it here with a red laser 647 detector, and I still remove quite a bit of autofluorescence. So if you don't have any other option, just use any channel. Okay, I'm still trying to find a very good example for autofluorescence. Try to see if I can find anything there. I'm sorry, I don't have anything on hand to show you. Okay. So yeah, don't hesitate to try Autospill. Really, I think that's the future of composition, way easier first to run, give you more accurate composition. So so try it, and any comment or any feedback, we're happy to hear it. Oh, Martin, you raise your hand. Go ahead, you can talk now. Martin, no? Okay. Carolina, go ahead.
Yeah, thank you. Just one question, you have finished? No.
Yes. Yes.
Okay. No, I have some questions. It was I'm using for the negative for the live date. I'm using amine bits, and I just realized that the negative for the amine bit is quite positive. So if I have, I mean, it's 10 to the 3 usually, and I just heard that it's not good to have a compensation when you have the negative so high. So what do you recommend, in fact?
So you're saying
It's really an issue?
So you're saying your negative is a 10 to the 3rd, and your positive
is 10 to the 6, more or less 10 to the 5.
Oh, your negative is
it's
10
not
to the
very
5?
High. No, the negative is 10 to the 3, and the positive is 10 to the 5. So the log is just 2 logs, so it's not too high. So look at here, the positive 10 to the 4th, fine, a little bit more, and put the negative 10 to the 3.
Okay.
There,
yeah,
exactly.
Yeah, you know, okay, so old school for cytometries, we told you to, we may told you to put it your negative at between 0 to 10 to the 2, but that's old school. We don't do that anymore. We want to make sure that you're above the noise of your instrument, and that's the 2.5 times the standard deviation of your electronic noise of the instrument. So if you know your instrument characterization, you can know that number pretty easily. If you have a BD instrument, the CST will give you that number. So as long as your negative is above that number, doesn't matter where they are. If your positive are still within the linear range, and within the linear range, same thing depend on the instrument, but for
Symphony.
BD, for the symphony, so symphony is 200 and I think 230,000, something like that, MFI. So as long as your positive is still within the linear range, that is because you are 10 to the 5th, so there's no problem there. So you are at 100,000, and your negative is above the noise. That's fine. Don't touch it.
That's fine. If with the RSD with 86, so that is not an issue. Okay.
Yeah. If you decrease the voltage, or it's not gonna change anything. It's not gonna make the separation better, or it's not gonna make your composition better. Nothing. So just leave it the way it is.
If I decrease the positive, in fact, the spread matter is bigger. So I prefer to have more or less of the positive over 10 to the 4th, between 10 to the 4th, 10 to the 5.
Yeah, no problem for composition.
Okay. Just I was asking what type of bit you were using. Is because in fact there are quite huge difference between BD and some like Thermo Fisher, where they have the positive and negative in the same. How do you resolve the issue of the Thermo Fisher give a high autofluorescence in BUV 786?
For the beads, you mean?
Yeah.
The beads give a high autofluorescence.
In that channel.
But the autofluorescence between the negative and positive doesn't matter for compensation, because
Okay.
you know because if you have autofluorescence is taken consideration for the calculation, because it's the same
Yeah.
autofluorescence and it's just the fluorescence of the dye that we're looking at. So it doesn't matter the autofluorescence there. I don't know. The only issue is just you're gonna have everything shift and you may not have a clear population. But cannot talk about those beads because I don't know them much.
Mm-hmm. And then my question was how you define a very good compensation to some extent, because I have done the compensation with the beads. I try also to do with cells. And finally, anyway, at the end, I have to do some kind of manual compensation when you do your X by X plots. And I mean, now I'm going to do with the autofluorescence to look if finally just without the manual compensation, I can reach the good compensation. But how do you define a good compensation? In fact, I mean,
That's a big question, and there's no clear answer. The only answer I can give you is look at your population of interest and look at the N by N and see if there's anything that looks weird. And there's no, I know it's not, maybe not the answer that you want to hear, but right now there's no algorithm or anything that can tell you this compensation is great, this compensation is not, and it's just by the biology that we can tell, okay, here doesn't make sense or here makes sense. I will say with Autospill, we have way less manual adjustment to do. So far, I've been using Autospill for a few months and I never had to manually adjust my composition since then for the case I had to work on. So when doing the traditional, there was quite often that I had to adjust manually the composition. So.
Yeah. Okay. So it's as far I well understood. I mean, you suggest anyway to use the spill compensation with beads or and just the spectral with the cells.
No, I will if you can. Yeah, if you can use cells on everything, I will do Autospill. I will say if you use beads on Autospill, you will be pretty close to traditional, just faster to do it because you don't have to define your negative and positive population. But the number will not be as accurate, I will say, if you were using cells as composition.
Okay. Okay.
That will just, if you use Autospill with beads, you will get the same numbers as traditional or really close. So still better because it's faster, but and you can remove autofluorescence with traditional, you cannot, but you will not have a much improvement in your compensation calculation.
Okay.
Then if you look at that slide that I have from another presentation, you see that cell. So here the cells, it's really easy to find the slope because this used not be a good compensation control because the positive population was hard to define, and or depending on where you put the gate, your MFI was varying a lot. But now with Autospill, because we find the slope really easily with this type of sample, that's becoming now a good composition control with Autospill. So with beads, it's just gonna do one positive here, one positive there. And I think I have a slide for the positive. Yeah, so that's but one positive, one negative. So the slope, you don't have really much slope there. It just it's gonna be quite a line there. Okay.
Yeah. You mentioned, I don't know if somebody is asking questions.
So far,
can I ask?
So
can
go
I?
ahead. Yeah, go.
Yeah, you mentioned, but because it happened recently, I had a 8 colors compensation, and then I was using the same compensation for 3 color. Why if I don't have some channels anyway, why I need just for the compensation to use the 3 color? I mean, if it was,
Sorry.
sorry, I did with the color compensation because
So you
it's the
do
day
8
that I
color.
had all the compensation.
Okay.
And and another day, I just stained the cells just for 3 colors, but I used the same 8 color compensation and didn't work.
Yeah, because you don't have the same spectrum. Because you have other color you have other color that's spilling into that you have other fluorochrome that's gonna affect the other signal. So even on different day, even if you use the same panel, so even if you use the same 8 color on a different day, because your instrument will change, maybe because your pipetting on everything change, you didn't pipette exactly the same quantity of antibody, and so your composition will vary day by day. That's why we always say you have to run your composition for every experiment and not not once and use the same composition for the rest. So composition has to be run every time.
Okay. But this what I'm yeah, but if you have beads, I mean the cells are always, I mean the beads, I mean if you do the compensation with beads, they will not change anything if you repeat every week.
Yes, your
or
instrument
once a month.
Your instrument will change. Your instrument change every day. Every time you turn on the machine, your instrument change a little bit. You try to compensate that. You try to avoid that by running QC on your instrument, but still, that's why when you run CST, your PMT voltage are getting adjusted every day. And if you don't adjust, if you put the same voltage so if you recall an experiment and just put, I don't know, 500 volt, for example, for your FITC every day, you're gonna see a variation of signal of your FITC day after day because you didn't take in consideration that your instrument changed over time. So that's why when you run CST, one day you may be at 500, the next day you might be at 505, the next day you might be at 492, etc., etc. Because the instrument is going to try to adjust itself to minimize this variation. It doesn't mean, even if we minimize, doesn't mean you don't have it. There's two questions on the chat. When you select beads as universal negative control, does it mean that you flow beads only as one of the symbol? Yes, Luran, that's why my universal negative beads were just beads non-stained that was run on the machine. That was 5, I think, if I remember correctly, 5,000 cells that was run, and there was no staining on it. And is it okay to use the same matrix on sample from a different experiment? Same panel, same voltage. So if it's on the same day, on the same machine, and you use the same color, yes, but that's a lot of things that need to be the same to be able for you to be able to do it. So in general, when you run on the machine, you're gonna run one day and after the next day. So you have to rerun the composition every time. And even when I'm saying same day, if someone shut down the machine or something happened on the machine and the machine has to be recalibrated, it won't be the same. So you're gonna have to rerun as well the composition. You have a lot of variation. You have variation on the experiment size where your pipetting and everything change. On you fix, you perm your cells, and you leave your cells for an hour in the fridge or 2 hours in the fridge. That will also affect your fluorescence of your cell. And on the other side, you have the instrument aspect where your instrument can vary over time because when turn on and off, you're not gonna have the same laser power, or your PMT may change a little bit. So good practice, always run your composition every time. If you did not use beads only, what's the alternative method to do for the issue? So if you're talking about composition, you can use a mix between composition beads and cells. On the composition, there's no problem there. When we define our composition, as I show you here, I can define different negative and different positive. I can say my 647, for example, was cells, so I can put my instance cells here, and for my positive in traditional. If it was on Autospill, I don't even have to define my negative, so you just need to make sure my cleanup gate is on my cells and accurately on my cells. So, and for the if you don't have a universal negative on cells, I show you on the traditional that you can set up a negative gate here on area D to define that negative. So instead of using the universal negative cells, which is here, I define a negative gate on my actual sample to say those are negative for that marker, and those are positive for that marker. So do you have to produce new control for every experiment, or what do you mean by redo the composition for every experiment? So both. So yes and yes. So you have to redo your composition control for every experiment, for sure, because your pipetting may change between every experiment. You may not use the same pipette, so when you think you take 5 microliter, maybe in one pipette, it might be only 4.8, on the other one, it might be 5.2. If it's on the same day and it's the same sample, for example, if you start running a sample in the morning and you run out of time on the machine and you're running the same experiment in the afternoon, your composition, because you left your sample maybe in the fridge or whatever, your fluorescence might have bleached a little bit, so your composition will have changed. So you're gonna have to rerun the same. You have to run the same control again. So because you run your composition, you do your composition control at the same time as your experimental control, and you run both of them together. So if your experimental control waited 2 hours in the fridge, and your composition control should have waited 2 hours also in the fridge before being run. Same thing, same condition. Thank you, Amy. Thank you, CK. So I'm just gonna put back my email address here. Thank you, Carolina. I'm gonna put by my email address, nicolas.love@pd.com. So don't hesitate to reach out to me if you have any question. You didn't have time to ask it today. If you need immediate support for Flojo, as I might be busy either at conference or trainings some sites, you can email flojosupport@bd.com. It's also the same email if you have any issue with your software or installation or something like that. Okay, so it's the 2 email address to remember. Thank you, Josh. Thank you, Pierre. Thank you, ACD. Have a good rest of your day. Yeah, Luron, don't hesitate to reach out to me. Just shoot me an email. Thank you, everyone. Bye.